I do have it in my title, but my title is there. But I left out a very important component as well, is that there's a telemetry component to this study as well. Okay, so the title is Genetic Connectivity Among Estuarine Populations of River Bream, Acanthopargus berda, along the southern African coast. This is part of my PhD studies. It's done in conjunction with Paul Cowley, Scotty Carl, and then Paulette Bloomer. Paul Cowley from SciAp, Scotty Carl is here, and then Paulette Bloomer from the University of Pretoria, Department of Genetics. Okay, just a quick overview of the talk. I'm going to introduce the focus area to you. Why is it important that we want to look at the Acanthopargus berda? The aim of the study, the markers and the methods used, the results and discussion, conclusion, and then some recommendations. Okay, this is the Cozy Lake system. I'm sure that everybody sitting here visited this site at least once. It consists out of basically 4 lakes. It's Lake 1 to large Lake 3 and Lake 4. The system opens to the sea just next to the mouth. Okay, this is the aerial photograph of the area. It looks really beautiful. If you go a little bit closer, it still looks beautiful, but there's something happening over there. I don't know if you can see it over there. It's traditional fish traps that is scattered throughout mainly the first and second lake, and then specifically in the mouth area. That is a huge concern at this stage. And the link between the first lake and the second lake here is only allowed through that 20-meter passage, around about 20 meters. Okay. So the problem is not just finding that little gap, but it's staying out of this as well on your way to move through the system. This is Acanthopargus berda. Some people might differ from what I'm going to say because there's another species and there's still some controversy about the naming of these species. There's only a single morphological difference between Acanthopargus vagus, a South African endemic, and Acanthopargus berda. I'm going to stick to berda because all the published data refers to it as Acanthopargus berda. Okay, distribution, northwards from East London, up into Mozambique, into the Indian Ocean, as far as Japan and Australia. Mostly confined to estuaries and river mouths. Sexual maturity is reached at a size of 25 centimeters. This takes approximately 5 years, and that's one of the problems for this species. Maximum size recorded 75 centimeters, very long time ago. And then winter spawning takes place near the estuary mouth. So what was believed to happen is that individuals from the system will migrate towards the mouth area, and then they would spawn, and then they would move back into the system. And then also the juveniles would move into the system. Okay, the study aim consists out of 2 components. It's the genetic component. We wanted to measure genetic variability and how is this genetic variability distributed within the sampling localities along the southern African coast. I made use of a mitochondrial DNA marker, which is maternally inherited. That gives you a historical view of the population history, and then microsatellite markers are nuclear markers, and that gives you a more recent view of the population. And then also a large component was the telemetry component, and that measured the movement patterns within the Cozy Bay system. Okay, so what I did for mitochondrial DNA, I used the ND2 gene, amplified 100 base per fragment, and then constructed an allele network. I'm not going to put in the details of the analysis programs that I used. If you want some more information around that, you can please come and talk to me. The microsatellites, we developed species-specific markers, and we amplified and analyzed 10 of those. Okay, so jumping into the results, I'm going to try and explain this to everyone. I'm not sure how many of you do understand this at first glance. This is the sampling localities, the sample sizes per localities in brackets there. It's low for Inyaka Island. This is what it looks like on the Southern African map. Each of these little circles over here represents a specific unique DNA sequence. A line between the different circles represents mutational steps, and in the case where we have dark circles, those are unsampled haplotypes. So that means that between this haplotype and that haplotype, there's 1, 2, 3 mutational differences. So it's further separated from this haplotype than that one, for instance. Okay, so in total I analyzed 45 randomly selected samples. Final fragment length analyzed 816 bases, 19 polymorphic sites, identified 24 haplotypes with one very common haplotype, haplotype 1, contained 19 of the 45 individuals, and that haplotype occurred all along the coast, so from each of those sampling localities. Nucleotide diversity, 0.25%. Haplotype diversity, relatively low at 0.72. And then just the basic layout of this network presents a history of population expansion. And this can be due to the fact that the population contracted and expanded in the past. Okay, because it doesn't look like it's expanding currently. Just in terms of the AMOVA, all the variation was identified from each sampling locality, so no variation between sampling localities. In terms of the microsatellites used, this just represents the number of individuals used per microsatellite marker, the number of alleles identified, observed heterozygosity, expected heterozygosity, and FIS. Now, this doesn't really mean anything to someone that's not a geneticist or a molecular ecologist. I just want to mention that I excluded 3 of these markers because of the high FIS value. It's an inbreeding signal, so that just basically gives you the same signal as what you would expect for another random marker. And then also these 2 markers seem to be linked or caused linkage between other markers, so I also removed those. So it left me with 7 markers. Now once again, those are the sample sizes and the sampling localities, what it looks like around the coast. Pairwise FST values, that gives you a signal of population structuring. Nothing really happening except between St. Lucia and Ungazana. There's a very low FST value, and that was significant. I'm still not 100% sure what happened there. I need to mention that these samples collected from St. Lucia was collected while the mouth was closed, so that might have an influence on the results. I will definitely further investigate that. And then just structure analysis. It's grouping samples that's more closely related to each other. It indicated that there's one large population, and then using another grouping method, you can see that there's some type of grouping taking place, but it's not really defined. So most of the yellow samples will be close together, most of the blue ones will be close together, but they are still overlapping. So the results here indicate that there is movement between the different sampling localities. Okay, the telemetry component of the study, we, as I said, focused on KwaZulu area. We deployed 8 receivers. Those are the deployment sites, so that we can track the movement of the fish through the system. It's a very nice area to do this kind of study because the fish needs to move through those narrow channels. So you are definitely, if it moves through there, you are definitely going to pick it up. Okay, and then we had to insert the transmitters into the fish. So we anesthetized the fish, opened up 2 centimeters, insert the transmitter, we'd close it up, and then we would make sure that the fish is okay before we let it go again. I have to mention that initially we planned to only sample individuals, 30 in total from Lake 3 over there, so that we can track the movement and then back again. That didn't work like that because we could only sample one individual from here on the initial trip. So then we arranged, it's not a competition, it's with the local ski boat club to come and help collecting the sample. We did offer a prize money of 1,000 Rand for the person that could give us the most fish to work with. We sampled in total 1 fish for the day. So that was definitely the most expensive fish that I ever bought, and I had to let it go again because it's part of the study. Okay, so this is all in total 22. We managed to catch 22 fish. So what we did after the study in Lake 3 didn't work, we started moving to the traditional fish trappers and bought some of the fish from them that were large enough to insert these transmitters. So in total we had 22 individuals at the end. I don't expect you to read through all of this. It's just the tag ID, the total length of the individual when we inserted the tag, the location where it was tagged, the date of first detection, the total number of detections for each individual, the station number, where it was first detected, when it was last detected, the station of first detection, station of last detection, and then the number of days that we were able to follow the movement of that individual. The samples highlighted in blue over here are all the individuals that we managed to track for longer than 344 days, or 331 days, longer than that. So those were quite nice to look at the results. And all the ones with the stars will be the individuals that we received the tags from. We had a reward system, so if the local people managed to find a tag, they could bring it to the office and they would get paid for that tag. Okay, and then unfortunately, we had 3 individuals that we didn't tag for longer than a few hours. Now, one of those individuals were caught later on, and the other 2 just disappeared. It could have happened that they were more vulnerable due to the operation, maybe, to predators, or that they died because of the insertion. Okay, this is just a little, very plain animation of what happens generally. The fish would move from where it was scored. If it was scored in Lake 3, it took around about 2 days to move to the next lake. And then from Lake 2, it could move to Lake 3, spend 1 or 2 days there, and then it would normally move towards the mouth, which it is not doing now. Okay, so it moves towards the mouth, spends 3 days in the mouth area, and then it moves back in a single day. Some individuals move from Lake 2 to the mouth area, spend some time there, 3 days, and then they would move back to the original area. So that was really surprising because the movement of the individuals also corresponded to the lunar cycles. So those are just information for 3 of the individuals, the days that they leave the location where they spend most of their time in, and when they would return to that location. And this is the new moon dates and the first quarter. So they would leave their location during new moon, and then they would return first quarter. And this happened from April up to August. And then just something interesting as well, for one individual with the highest high tide, most of the activity was inside the mouth area, and then as soon as the tides get lower again, they would move away. Now, this specific individual, we do not know whether it moved into an area between the mouth and the first leg, where it remained out of sight for the receivers, or whether it actually left the system into the sea, because between August of this 2010 and April of 2011, it spent somewhere which we don't know. So that could have been outside in the marine environment, moving from one estuary to another estuary. That's a possibility. Okay, so in conclusion, the mitochondrial DNA ND2 result showed signals of high gene flow historically between the different locations, between different estuaries along the coast, which wasn't really expected because it's seen that this species uses a single estuary where it just moves towards the mouth and then spawn moves back into the system. The microsatellites also showed high gene flow, but a weak signal of population structuring. So there is definitely something telling the fish to move back to its original location with only some limited amount of gene flow between the different estuaries. And then the telemetry, a high level of movement in the system, more than expected, because it's not a single migration towards the mouth and back. It's a monthly migration, depending on the moon, where it moves every month to the mouth and back for a period between April and August. And then also a lot of scope for potentially leaving the system and returning later to the system again. And then just a few recommendations. Active management of the traditional fish traps. I know it's easier said than done, but the problem is that the number of fish traps are actually increasing, and then also it's not evenly distributed. That would help. Restricting the size of a specific trap, and then also the number of fish baskets per trap would also help. Monitoring and removal of illegal building material. In the past, they could only use traditional or local material to build these traps, and nowadays they are using nylon ropes and other types of material to build that makes it more effective in catching smaller fish. So that's a huge problem at this stage. Preventing construction directly in and around the inlets, so that is relatively taken care of. To re-establish a fish joint management committee. Those are local people in conjunction with people with authority to try and manage the building of these traps, and then also to collect all the fish trap catch information. Okay, and then also patrol and removal of gill nets, especially in the narrow channels. Now, apparently this happens during nighttime, where there's lots of gill nets in the system, and then they just remove it early in the morning again. And then also re-establishing a working catch card system for recreational fishes as well, so that that information can also be collected. Thank you.
Thanks very much, Carl. Time for questions. Bruce.
Thank you. Hi, Carl. A two-part question. The first one, in your genetic analysis, and I don't understand it well enough to be able to understand, did you detect enough difference to suggest that we do have two different species? I mean, you had a small sample size, but could there be two species in our systems, which is what we believe there is currently? And then the second question is, there's a hell of a lot of information about this fish in KwaZulu, and the work of Scotty and Pat Garrett kind of has really given us a very clear idea of what they're doing. I think what the telemetry has added is that they're moving down there and then coming back. But we know that the fish move down. They're all males in the mouth area, and that's where the spawning aggregation occurs, and the females come down to join them. When you put the telemetry tags in, did you check their sex?
Okay, so I'm going to answer the second question first. Yes, so we did tag those that were ripe and running, we could determine the sex for. There were a few very small individuals that we also tagged, which we couldn't determine the sex for in the mouth area itself. But most of the individuals we did find males and females. And then in terms of the signals for detecting two species, what we would expect to see is this location, the Sundays estuary, is outside the range for Acanthopargus berda. So we would assume that these samples would be Acanthopargus vagus at least. But when we include all of them into the analysis, it's as if it's one species. You would expect something like this, for instance, where you would find all the samples for Sundays estuary on this side with lots of mutational differences between the two.
Thank you.
And what I forgot to mention, can I just mention this? You will also see that lots of the variation is actually within St. Lucia and Cozy Bay. I didn't mention it, it's extremely important. You will see that lots of different haplotypes occurs only in St. Lucia and only in Cozy Bay. So that's also a signal that Cozy Bay might be extremely important in housing the diversity for the species.
Any other questions? If not, thank you very much, Carel.